Bon, bonjour tout le monde, on va démarrer. On avait un petit débat rapidement parce que souvent nos speakers ils viennent, ils sont anglophones donc on se pose pas trop la question, on fait les talks en anglais. Mais aujourd'hui on a un speaker francophone.
Salut, comment ça va?
Mais je pense qu'on va malgré tout faire le talk en anglais, la raison étant qu'on a des étudiants étrangers, on a cette chance d'attirer des étudiants internationaux ici et que ça les coupe des talks et c'est aussi un excellent exercice finalement. C'est un excellent exercice.
Est-ce qu'il y a des gens qui se sentent, enfin, qui préfèrent vraiment que ce soit en français? Non. Ok. Est-ce que
et si c'est le cas, n'hésitez pas à venir me voir après le séminaire. Je sais que c'est toujours un point un petit peu complexe parce qu'on est dans une université francophone, dans un établissement très francophone. Et moi, j'adore le français, tout ça, hein, j'ai pas de problème avec le français. C'est juste pour les explications de données dans un contexte scientifique où l'usage de l'anglais fait du sens.
Surtout s'il y a des gens qui ne pourront pas suivre.
Il y a des gens qui ne pourront pas suivre dans les faits. Ok, donc...
Est-ce qu'il y a des gens qui n'ont rien compris à ce qu'on a dit jusqu'à maintenant?
Il y a ceux qui peuvent pas souvent en français, en anglais, non. Il y a aussi des gens qui ne peuvent pas suivre en anglais.
C'est pour ça qu'on a posé la question et que si c'est...
Est-ce qu'en français, par exemple, pour vous, ça serait mieux?
Moi, j'étais contente de savoir que c'est en français, mais je peux suivre en anglais.
Okay. Parce que l'autre option, comme j'ai dit, c'est j'ai les diapos en anglais, je peux parler en français, mais...
Okay. Bon,
enfin, mais
je propose de faire celui-ci en anglais une fois de plus.
I can do it in 2 languages, okay. English and French.
That's actually, if you want to turn into a Quebec politician later on, it's a good skill to have. Okay, so I'm very glad to welcome Karim today. You know, obviously, Karim is now our official UNF seminar organizer, so definitely you've very likely met him. Karim is prof, psychology department. He's an expert in electrophysiological signals. He's also co-director of the magnetoencephalography system we have on campus. And he's been with the center since you basically moved in Montreal, right?
Yeah, 4.5 years ago.
And recently he started being a bit more active because he has joined UNF. So UNF is mostly an MRI scanner, but we have a number of other devices, including an EEG that we're going to activate. Well, it's really Julie Carrier and Julien Doyon's EEG that we're going to try to make available for the wider community. He's also very active in everything related to machine learning, and he's going to sort of help with those aspects as well in the team. So it's only been a few months actually that Karim joined the UNF team, yep. But I'm absolutely delighted he did. It's a lot of fun to work with him. For those who have had the chance of working with him, you also know that he has this sort of like ADHD behavior, which means that lots gets done, and that's wonderful. That
yep.
really is wonderful.
Absolutely.
So,
I don't know what you're talking about.
So yeah, we everybody at the center felt it was important to have sort of like proper official welcome for Karim, so that's why we invited him to give a talk today. Thanks so much.
Thank you so much. Merci Pierre. I have been told to make just one quick comment before we begin. Arnaud Bourré, who's over there, just told me to remind everybody there's a new system for the registration for the MRIs. So if you wanna sign up for your MRI experiments using the new system. If you have questions about that, you can talk to Pierre, but also to Arnaud, who's over there in the back. Donc si vous avez des manips IRM, il y a un nouveau système d'inscription pour votre site.
Donc il faut aller parler à Arnaud et André.
Arnaud, et principalement André.
Et sinon sur Slack aussi, pour ceux qui utilisent ce système-là, vous pouvez poser vos questions là-bas et puis on fera le suivi.
Ok, donc ça c'est pas mal. Peut-être oui, good idea. Okay, alors, so basically here's the overview of the presentation. I'll give a brief introduction, and then the idea was really to talk about machine learning, but it's like a sort of a machine learning 101. This came out of the discussions I had at the beginning, including with Pierre, but also from the very beginning of talking about the idea of giving this talk, Sébastien also thought it would be a good idea to have a brief overview of what is machine learning, also aimed to people who have never used machine learning before. Also, I was thinking that because it's CRIUGM, we might have some people interested in clinical applications or come from the clinical side of things who have heard about machine learning but have not used it yet. So this means that for many of you whom I recognize, this is going to be quite boring at the beginning because there's a lot of things that you already know. For some others, it's hopefully going to be useful because it's going to give you some of the basics of machine learning, and for some others, it's just going to be maybe a useful reminder. And then I'll give you some examples, some take-home message and an announcement. Ça, le computer RVB, ça va rester tout le temps? Normally it should, hein? Bon.
Yeah.
Bon, j'espère que j'ai rien en dessous d'important. So, first of all, some nomenclature, and this is really important. It's always quite messy. If you're not familiar with the field and you're following what people are telling you over the media, you're going to hear about things like AlphaGo, Google's AlphaGo AI beating the world best player in Go. And then it's never really quite clear from the articles you see, is it AI that did it? Is it machine learning that beat this guy, or is it deep learning, and what's the relationship between these things? And it's often quite fuzzy, and one of the reasons is also because journalists don't really know what they're talking about. So they just use these words interchangeably. And so I think it's really important to start with setting the scene. The bigger picture is AI, artificial intelligence, and this is a field that dates back to a long time ago, back to the 50s, where the idea was to try to mimic human intelligence or brain processing with machines, AI with computers. Within this field of AI, there's another subfield. It's not the only thing that AI is, but there's a subfield which we call machine learning. And typically this is an algorithm that's going to be sorting out your emails and telling you this is probably most likely to be spam, and probably just throw them directly into your spam folder. And sometimes you need to go check if everything really is spam. Most of the time it's doing quite a good job. This started also a long time ago. And then within the field of machine learning, there is a subfield which is called deep learning, which has now received a lot of attention over the last couple of years, mainly for an increase in computational power, but also the availability of huge data sets and access to a lot of data. So it's important to keep in mind the hierarchy between these things. There are a number of things that, for example, belong to AI that are not necessarily classified as machine learning. For example, some would argue that a lot of computational models that we use in neuroscience, training models, doing predictions with models, this is probably also in the realm of artificial intelligence, not necessarily a machine learning algorithm. And so to link these topics together, again, artificial intelligence is here, within it machine learning, inside this deep learning, and this is related to a number of other concepts that you have heard a lot. We talk a lot about big data, data science, data mining. These are all interrelated, and to make this even a bit more complex, this is the one that's getting a lot of attention and is also very confusing. What belongs to data science and what doesn't? And many things pertain to data science, for example, statistics, using maths, a hacker mindset, visualization of data, advanced computing, signal processing, data engineering. All of this is somehow related to, in one way or another, to data science. And actually, to be honest, data science has become also something that means everything and nothing. So today, if you're looking for a job, you go onto LinkedIn and you put under your name data scientist. Nobody really knows what that is, but then people will contact you because they're interested in hiring data scientists, even if they, basically for a lot of people, if you're manipulating data, you probably are a data scientist. Yep.
Just a hacker mindset.
Yeah. What it is, or yes. Well, basically, it's not the typical science methodology. This is why. So, the scientific method is maybe based on academia, of ways of applying methods to and hypothesis-driven research, or applying methods to extract information from your data. A hacker mindset is probably sometimes, when you go to hackathons, for example, you'll meet people that don't come from academia necessarily, but they know how to code, or they will be thinking outside the box and say, well, maybe this other technique used in this other field, let's try and change it, manipulate it, hack it, and see if it can give us something interesting in this field. So basically, it's also sort of a cross-fertilization across different fields. So what I'm going to talk to you about today is data-driven neuroscience. And the idea here is that more and more we're seeing opportunities to use brain decoding, using tools that are borrowed or come from the field of AI, as a powerful tool for neuroscience research. And I'll try to explain this in a bit more detail of how this can be done. The idea is that you record brain signals, There are many ways you can do this. Of course, you can use fMRI, you can use EEG, you can use MEG, and you can use FNIRS and others. This gives you a set of signals. And then out of this, there are a number of things that you can consider doing. You can explore this, see this as a data mining question, and the questions you may ask is, what features are going to be useful for me? What cognitive processes I will be able to tackle? Are there any brain dysfunctions that I can try to use this brain data mining approach to try and explore in more detail? And this is typically what we do in the COCO lab. This is my lab, the Computational and Cognitive Neuroscience lab. This picture is a bit outdated. We're a bit more now. So basically what we do, we record brain signals, MEG, EEG, or intracranial EEG. Based on that, we run some signal processing techniques. This is spectral analysis, for example. Then we use machine learning to do a number of things. We can decode or classify or predict, depending on your question, a number of things. I'm just going to give you an overview of some of the topics that we're tackling. Decisions, motor intentions. We have a project where we look at different styles of meditation or different types of meditation. Sleep stages, this is something that is useful because we can automatically classify sleep stages. This is something very helpful if you're working in a polysomnography, with polysomnography data, for example, in a sleep lab. We have an ongoing study I will show you a bit more about later on, where we're trying to see, do brain signals from people that recall their dreams differ from people that say they don't recall their dreams? Are there any changes in brain activity that we can identify using machine learning? Obviously, you can use machine learning as a person identifier. Can we use EEG signals to identify who you are? You can also tackle brain disorders. I'll probably talk about schizophrenia. And we have also memory, episodic memory tasks that we're using this for. So I will come back to some very specific examples afterwards. I'm just now going to go through some basics of machine learning, and this is the part where some of you can switch their brains off for a while, because it's, probably you know this. So at its basic, it's the practice of using algorithms to parse data, learn from it, and then make a determination or a prediction about something in the world. So basically what happens here is that the machine is trained using large amounts of data. Large is obviously very relative adjective, depends on your field. Some big numbers for another field are going to be small number for another field. We can come back to that later. And then algorithms that give you the ability to learn how to perform the task. And so I'd like to first start with the first important distinction. Many of you have heard about deep learning. In contrast to what some call shallow learning, the distinction here is that shallow learning often refers to traditional or conventional machine learning pipelines. You have all heard of methods such as SVM, maybe, or LDAs, linear discriminant analysis. So the idea here is that you handcraft your features, so you extract your features from your data. You have some a priori knowledge that tells you that these features are probably important for this task. Features, the French word for that is attribut. So you compute them from your data, and then that's what you're going to use to train your algorithm to make some predictions or to make some classifications. By contrast, in the case of deep learning, it often refers to lengthy and more computationally expensive machine learning pipeline, where you do the same time feature learning and model training. So you are learning representations of your data and training a model at the same time. So the features are actually derived from the data, and this is what we call representation learning. And you can see here two examples of neural networks. The deep actually refers to the fact that you have all these multiple hidden layers between your input and your output. A simple neural network would look like this. So you have artificial neurons in, and then you have your output vector. So you have your input layer and your output layer. Another way of framing this is in the shallow learning, you have your input and you extract your features. First step, second step, you're going to run some classification based on these features, and then it's going to give you an output, tell you, is this a car or not a car? In the deep learning, you only have one step where you're doing feature extraction and classification, and you have an output. Now, there are a lot of questions about what interpretations can we make from the features, the way they have been automatically generated within these layers. And obviously, they're not as easy to understand compared to the ones that you've extracted yourself based on domain knowledge. It doesn't mean that you cannot use them, but they're just a bit more tricky to handle. Okay, now the second distinction that is important, supervised versus unsupervised. Supervised learning is where your data is labeled, so you have classes, apprentissage supervisé. So you have classes, you have your different labels, so des étiquettes, and this tells you, for example, that you have images of dogs and cats. and you know which ones are the images of dogs and the cats, and that's what you're going to give to your algorithm, and he's going to use that to learn and try to be good at distinguishing dogs and cats in the future. But you actually gave him the answer for the data you've given him. So the training comes also with the cheat sheet, because you've given him the answers. Whereas the unsupervised learning, in this case, the data has no labels. And so your classes or your categories of the data will be determined by the algorithm from the data. I'll come back to some examples of this later on. So now, let's go one step back and go to the basics of the very basic supervised learning approach. And we're going to keep it very simple. A two-class decoding problem. Binary classification, let's assume we have two cognitive states. For example, attentive and distracted. Okay? State -1 and state 1, or state A and B. So what you do, you have your brain signals, and then you compute a feature vector, or vector d'attribut. And there's a number of steps that I have skipped here. We can discuss them a bit later, like how do we extract the features and how do we select them best. Once you have these features, you're going to classify, and your algorithm is going to tell you, okay, this epoch of data, or this segment of data, corresponds to state 1, or cognitive state A, or cognitive state B, for example, attentive or distracted. And what's the basic idea behind this, behind specifically supervised learning, is that you have a set of data you call the training data. With this, you're going to train your classifier. In other words, you're actually training a model, right? Then you have test set that has not been used to train your classifier or to create a classifier. This is really important. It has to be naive. And then based on this test set and the application of this classifier on this test set, again, the classifier was only trained on this data over here, had never seen this data. This is why we call this a test set. This is going to give you some predictions. And the accuracy of the predictions is it's going to tell you how well your model or how well your classifier works or predicts your data. So let's go into a little bit more detail here. The learning process for the supervised learning can be formalized as a process by which you choose and you fine-tune the rules of a decision function, f. So this is going to look like this. You have your training set, you have your known labels, and we're searching for the parameters of a decision function f, which will allow you to correctly classify the other samples that come from the test set. Okay? And I'll make this a little bit more visual to simplify. So first of all, the fact that we train on training set, and then we try to apply this to test set, and we hope it's going to work, this is exactly what we refer to by generalization. In other words, your algorithm has trained on data where you actually gave him the labels. You say, these are dogs, these are cats, and it learned to do that. Afterwards, if you apply that to a new image and it correctly classifies, it means that your algorithm is capable of generalizing. Why generalizing? Because it's beyond what it has received as training data where the answers were given. Okay? It's able to make correct predictions on unseen data. And just to visualize this, we have two features, feature 1 and feature 2. And assuming we have two sets of data, you can see here in yellow and in blue. The process of learning, so the training process, is a process of actually trying to identify this decision function here, or this line here that separates your two data points, or the two labels, label -1 and 1. And then what happens is this new dot here is a data point that was not seen beforehand by your algorithm. Okay, your algorithm was based on the blue and yellow, and it learned that this decision function is probably quite good at separating them. It's obviously never this clear, but just for the sake of the example, I'm just showing this here. Now this new point comes in, and what is my algorithm going to predict for this red dot? Is it going to classify it as blue or as yellow? Blue. Absolutely. It puts it blue if it's correct. And why? It's because it happens to be on this side of the decision function. And so this is an example with just two features. Obviously, in most of the tasks that we're doing, we're using hundreds and hundreds of features, sometimes a bit less. But so it's an n-dimensional space where we're basically doing the same thing. So instead of training a line as here, you'll have a plane, or it could be a hyperplane if it's in n dimensions. Yeah.
It's not 100% sure. Like, is it, you have a statistical level?
Yes, we have performances metrics, and yes.
There is a level of chance.
Yes, absolutely, and I'll come to the performance metrics.
Okay.
So again, you have your brain data. Again, I'm summarizing for you the way we go through supervised learning. We have brain data and observations. You split this into training and test. you take the training, you train your classifier. There are many ways you can do this. I will not go into the details right now. And then, based on this, you run your classifier in your test set. So you have the training phase, the test phase, and the result that it got on the test set, so unseen data, this is what we will use to calculate the performance. And generally, one way of looking at the performance is the percent of correct classification. So if it managed to get 70 correct answers out of 100, then it's a 70% performance or correct performance rate. So decoding accuracy, DA, equals 70% in this case. Okay. Another important distinction, a third one to make, is between classification and regression. So typically, both can be used in a machine learning context. For the classification, these are discrete labels, as I said, cats and dogs, for instance. And in continuous values, these can be, for example, prices of cars, for instance. In one case, we'll be talking about classification, and in the other, it's a regression. Most of you are familiar with linear regression. You can also run regression in the context of machine learning, where you actually learn your regressor on a subset of the data, and then you look at how well it predicts on the data that you have left outside. And you can do this again also with cross-validation. So maybe I did not mention cross-validation. I'm going to go this is, I skipped this. Cross-validation is, as you've seen here, I've split the data into the training set and the test set. And then I look at the performance. But then I might get a good performance just because the way I split my data into training and test was actually lucky, right? And so I get a good performance. But how do I know that that's always going to be the case if I used another split of the way I split my data into training and test? So the way we deal with this is cross-validation, where we actually repeat this over and over again. Okay, so you have training and test, we look at the performance, and then we say, okay, now I'm going to take these as training, and I'm going to leave these others out as tests, and I see how well it performs. And I do that over and over again. There are different techniques of doing that. K-fold is a standard, leave one out, or the LOO method, L-O-O, is another one where you actually, for example, just leave one subject out and train on the others, and then you leave another subject out and train on the others. Different ways of doing that.
Make statistical analysis all cross-validation.
Yes, and then you average your decoding accuracy across all your folds.
And this is your overall accuracy.
Exactly, and you know that this one is not biased by just the way you split your data, because you split it in many different ways. Okay, so I mentioned classification and regression. So in regression, you have a continuous value, and so you predict actual values, whereas in classification, you have two classes. And so sometimes machine learning is presented using this discrimination over here: supervised machine learning, unsupervised, and then within the supervised you have classification and regression. In the unsupervised you have clustering and association. I'm not sure I kept the slides where I described the unsupervised, but some of the unsupervised techniques would be, for example, using k-means or hierarchical clustering, where you try to identify within your data clouds that separate within your data space. Then you compute the centroids of these clouds, and then afterwards, when new points come in, you just compute how far or how close they are. What is the centroid that is closest to this new data point? That's one way of classifying them based on that. Okay, so we come to the question of performance metrics. How do I know that my classifier is performing well or not? There are different ways of doing this. One is classification accuracy or decoding accuracy. This is the one we just mentioned. You can look at the logarithmic loss. You can look at the area under the curve. This is also very standard to use AOC. There's a confusion matrix that gives you useful information, and I'm going to show you some of this in a second. Classification report for regression metrics, it's R squared, mean squared error, and mean absolute error. I'll briefly discuss classification accuracies and the confusion matrix, just to give you an idea. So, have four classes. So these are the true classes. This is a confusion matrix. Assuming you have four classes that you want to separate with your machine learning algorithm. Okay, these could be, for instance, four clinical populations or four different brain states. Okay, so these are the true classes, and here you have the predicted class on the y-axis. So, what would a perfect predictor give you? If you have perfect prediction, what's the confusion matrix? I know nobody's asking, probably everybody thinks, well, this is obvious, we know confusion matrices, but if your classification is 100% correct all the time, what would this confusion matrix look like? I just need one word.
Red diagonal.
Thank you very much. So it will give you a red diagonal, meaning that true class 0 was always identified as class 0, class 1 as class 1, class 2 as class 2, and so on. But life not being that ideal, it's not always the case, and you're going to have cases like this, where your true class 1 has been most of the time identified as class 1. This is why it's yellow here. In like 75% of the cases, I can actually add the numbers, 65% of the cases. But in 10% of the cases, class 1 was identified, the predicted class was 0. And in another 25% of the cases, it was identified as class 2. Okay? Why is this useful? And why is it useful to look at your confusion matrix most of the time when you're doing this type of analysis? Because this is going to tell you, when my algorithm fails, why is it failing, and what classes are getting confused? Because when I see this, I know that my algorithm is really good at detecting state 0, brain state A0, also 2 and 3. I know it has a problem now with class 1. So if I'm looking for new features, or if I'm looking to try to improve its performance, this is probably where I should focus my attention. I need to try and make it better at identifying class 1. Okay? And I know that it never mixes class 1 with class 3. It actually has a problem with 0 and 2 being closest. Yes. Okay, so theoretical chance level, this comes back to the question we had before. 2 classes, if your algorithm is just doing n'importe quoi, it's going to give you 50%. 4 classes, who's quick? 8 classes? 16 classes? No, I'm not going to ask you that. So we need to be careful with theoretical chance levels, because they are theoretical chance levels. And I'm going to explain this in a second, a bit more detail. It's very easy to exceed your chance level just by chance. Why? It's because you can easily imagine if you have 10, if you have, let's say, a box with 10 balls in the box, right? Or let's say 20 balls in the box. 10 are red and 10 are blue. By chance, you can if you're going to take 10, by chance you can easily find yourself picking out 8 red ones and 2 blue ones, and you would not be surprised, right? You only have 20. So in that case, you can say, well, I've got 80% red and 20% blue, so probably the distribution there is 80-20. It's not at all the case, it's just that the sample size is really small. So the theoretical chance level doesn't make sense for these very small sample sizes. In other words, if you go beyond 50% in your machine learning result, right, but you only have, let's say, 30 observations, and if you find that you got 60%, is that something that you should be happy with and you go publish your paper? Or you say, well, 60% is not that high, I only have 30. So there is a very easy parametric and non-parametric approach to tackle this. The bottom line is that the answer to this is using statistics, of course. And so some people come to the field of machine learning thinking, okay, p-values is not working out for me, the t-test is not working, I'm just going to run an LDA. And if I have something beyond 50% in my 2 classes, then I'm going to publish that, because then it's going to solve the problem. I'm going to say my predictor is above chance level. And of course, it doesn't happen that way. There is no free lunch. So what you have to do, you still have to do statistics on your decoding accuracy. And you see whether your decoding accuracy is statistically significant. The most elegant way that I think this should be done is using non-parametric approaches, and the best way to do that is permutation tests. This is very, very simple. You had your 2 labels for your 2 classes. What you do, you look at the decoding that you get, and then you just mix your labels and rerun the decoder. So now you've given data to the decoder. Basically, there's no structure at all, okay? Because you've actually mixed the labels by chance. So dogs become cats and cats become dogs. And you give that to the classifier, and then it's going to try to do its best. And it might find 55% accuracy or 53. And then you're going to mix again, and you give him again to do that classification. So if you give him 1,000 classification tasks, all of them using this random mixing of your labels, then you'll get a null distribution of your decoding accuracies. And you'll see the values that they get. And you want to make sure that the one that you actually got on the real data happens to be at the 5% or the 1% of that nil distribution. We've discussed this in this paper if you're interested, but it's really very basic. And people doing machine learning and applying this know about the statistical approaches to tackle this. The reason we did a paper for this is that in the last years, were more and more people coming in from other fields, including neuroscience or neuroimaging, applying machine learning techniques, there have been a number of papers where people just bluntly published the decoding accuracies and thought that was the end of the story. But in many cases, they had very small sample numbers. So if you get a 60% with a 20 versus 20 population, then that 60% could be significant, but it could also be totally insignificant and just out there by chance. Okay. So what's the take-home message from that part is you can't work your way around statistics by switching to a classification framework. You will still have to do statistics, but in this case, not on the distributions, but actually on the decoding accuracy. And all classification metrics will require this statistical assessment. I recommend permutation tests. And comparison of means and machine learning actually are conceptually very different approaches. Some people try to compare them and say, well, I did statistics, I got this. I ran machine learning, I got this. It's not exactly the same thing. Okay? Conceptually, there are several differences, one of which are the idea of generalization. When you're doing a machine learning approach, you're actually trying to generalize to unseen data, and you want your results to hold for data that was unseen. That's the way you train your data. Whereas in standard statistics, most of the time you're using all your data. There is a couple of papers, this is one of them, by Danilo Bzdok, 2017, and he has one or two more that actually give a quite nice overview of the relationship between classical statistics and statistical learning in imaging neuroscience. Okay, that was a bit quick. So these are other things that I did not have time to talk about. We can talk about them more, but these are also other things that are happening right now that are quite exciting. Transfer learning and a related issue, which is domain adaptation. How do I adapt my data so that it fits between two different data sets? Transfer learning is when you train your algorithm on one type of problem, and then you take that trained algorithm and you try to apply it to a different but closely related type of problem. Sometimes that works, sometimes it doesn't work without some domain adaptation. In domain adaptation, you're going to adapt your original data to some of the distributions that come from the new data. Meta-learning or learning to learn is something that is of increasing importance. I didn't have time to get into any other details here for the deep learning and the adversarial learning, so there's much more to dig into, but the aim was really to just go back to the basics of simple machine learning.
Okay.
Yeah.
Can I ask you, in terms of the cross-validation method, you didn't mention the out-of-sample or the independent sample validation.
Yeah.
But that's...
Yeah, so you can split it into your training, validation, and your test set, depending on your problem. In some problems, you just have training and test. One of the things that is highly recommended is, for example, and this can relate to your question of validation and test, is doing nested cross-validation. In nested cross-validation, what you're going to do is you want to say you cut your data into training and test. Now you take the first fold of the training and you want to find the best algorithm that works for this before you apply it to the test. Now there are different ways that this can happen. So what you want to do, you have different parameters and you want to optimize your parameters, you want to choose the best features. So the best way of doing that is you take this training set and you again split it into a test and a training. And so now you train on a subset of the train, you test the performance of that model, and then do it over and over again until you get the best model. So this is a nested cross-validation. And so you could consider that also to be validation and test. Okay. So what I'm going to do now is two things. First of all, I'm going to present a list of examples. I'm not going to have time to go through all of them, but I just want you to take a look, and then afterwards, with a show of hands, you tell me what study you want me to talk about, and I'll present that to make this a bit more interactive, like the last Netflix. What's it called? No, Black Mirror. Yeah. So these are some studies that I have here slides for. We have others in the lab, but I'm just going to show you this for a second. So we have studies looking at schizophrenia using MEG and decoding, or decoding motor intentions, decoding decision-making processes, emotion detection, states of consciousness. This is anesthesia versus wakefulness, dreamers, non-dreamers, and a nice olfactory episodic memory task, where we train people to learn an association with referred to smells or of olfaction, and then afterwards, 2 days later, we try to see how well they can remember them. I'm not going to ask you right away to tell me what study you're most interested in. We'll come back to that later with a show of hands. I'm just going to take a 5-minute break to switch gears, and this is something that I thought could be of interest. I want to tell you about UNIQ, to make sure I've told you about this, and then afterwards we'll get back and I'll ask you what studies you'd prefer me to talk about, and I'll talk about 2 or 3 years, much time we have. Okay, so very briefly, I know many of you are already familiar with UNIQ. These are slides from a meeting we had recently with FRQNT. UNIQ is a research cluster, a regroupement de chercheurs, that has been very recently, about a couple of weeks ago, approved by FRQNT for funding for a first round of 2 years. And then after that, if we renew it, it's cycles of 6 years. And the idea is to create a new center where the idea is to unify neuroscience and AI in Quebec. And I'm going to quickly run you through the rationale. It's really just a few minutes. So the idea was that the future of research in AI and neuroscience both would benefit from a better synergy between the two fields. Quebec has a critical mass of researchers that are internationally recognized and well known for their research in neuroscience and in AI. So we have a very strong scientific and economic ecosystem for this. And so it's an ideal situation to bring in something like this where we actually bring the two fields together. It's a unique opportunity, of course. So the mission is, first of all, to promote new synergies between AI and neuroscience, and also to try and better understand biological and machine intelligence by looking at their common principles. So this is always a question where people say we want to do synergy between neuro and AI, or bring the fields together. And everybody says, well, this sounds very nice, and obviously people don't want to fund this, but how are you going to do this? How are you concretely going to bring these two fields together? And so the strategy we chose within UNIQ, and this is really the result of a lot of work between all the researchers involved, including many of the people that are here, was to say, okay, let's go back to some basic principles of what we call intelligence. For example, issues such as concepts such as learning, concepts such as neural networks, concepts such as higher order cognition, and concepts such as sensory or motor processing. These are things that are central to intelligence, be it from a machine perspective or from a human or biological perspective. So I'll get back to that in a second. We want to achieve a strong leadership in this new AI neuro field in Quebec and internationally. And the fourth mission is to really contribute to creating a new generation of students and professionals in the field of AI and neuroscience. And last but not least, we want to work strongly on improving EDI, so equity, diversity, and inclusion in the fields of AI and neuroscience. In AI, it really is a big problem, but I think it's also still a problem in neuroscience. We need much more gender balance, and we need much more inclusion and presence of minorities and so on. So this is something.
What is PHQ?
PHQ is highly qualified personnel hautement qualifié. So people like you would qualify by PHQ. Were you fishing for a compliment? You got it? Donc, la pertinence, why is this important? Because we have it's really important that we do this from a scientific context, because there are other places around the world, not so many, but some are starting to create such centers, including Stanford and MIT and London. So it's ideal now, and it's urgent to do this before the competition beats us, of course. So this is going to allow us to put a lot of things together, a lot of resources, both for imaging and also for computations and for data acquisition and collection. And so scientifically, we want to bring these two fields together, machine intelligence and biological intelligence. But the way we want to do this is actually by creating a fusion between the two, so integrating them into one and the same problem. To do this, we have different axes: perception and control, as I said, cognitive processes, learning, and networks. For which we have a number of researchers that are co-leading these different research axes. And also we have transversal task forces where we have platforms, neuroinformatics and data science for this. Also technological innovations, and a group work looking at neuroethics. 45 researchers across Quebec. 7 universities, UDM, McGill, Laval, Polytechnique, Sherbrooke, ETS, and Concordia, plus 31 members and collaborators from outside, from Quebec, but also internationally, which gives us a total of 76. You hear the breakdown. You can see in terms of number of researchers involved in UNIC. There are 18 research chairs, Canada, 9 Fs. These are a lot of numbers, but this was important for the meeting. We had to have our Q&T, maybe less important for you, or just to give you the idea of the breadth of UNIC. And so UNIC members are involved in different companies also. Many are involved in Element AI, but also we have a researcher that's in Google Brain, DeepMind, Microsoft Research, and Facebook, and so on and so forth. Organigramme, boring. Budget annuel, less boring. We have 809, well, basically the FRQNT pitches in with 300,000 a year for the 2 first years, and we managed to get at least promises of financial support from different apogées. Apogées are the CFREFs, so IVADO, Sentinel Nord, Institut Transmedtech, and HBHL, totaling 330,000 here, and 264,000 from the universities, so a lot of thousands. Gives us a total of roughly 900,000 a year, so we're roughly to 1.8 million for the first 2 years. And what is this going to be used for? This is like the big question, of course. This is going to be mainly used towards creating events, workshops, summer schools, and also seminars, international speakers coming in to talk to this AI and neuro, and then increasing synergies by providing scholarships. So scholarships for students, primarily docs and postdocs, probably also master's students, and also internships, with the requirements that they be supervised by an AI PI and a neuro PI. Some of us are already doing a little bit of both, but basically this is one key way we chose to try and enforce this and help increase the synergy by saying applicants need to identify, or their project needs to include 2 PIs from the 2 sides. EDI is a really important thing that we're looking for, and we'll have a committee that is dedicated to this, and they will propose some very specific actions that we can do to promote equity, diversity, and inclusion. So long term, we're hoping that UNIC is going to become a reference internationally for AI and neuroscience. Midterm, we hope to have a strong impact also on teaching and on EDI, and a strong leadership in Quebec and Canada. And very short term, we want to have a strong impact locally, and also actually get these things started by announcing in the next coming months the first call for scholarships and so on. So that was for UNIC. That was a quick diversion. I don't know how much time I have, probably not too much, so I'm going to show you the list again. Okay. So I'm just going to ask you, and raise your hand if this is what you'd like to hear about most. Okay. So schizophrenia. Okay, 1, 2. Motor intentions. Decision making. People are hungry actually. They want to go. Oh, okay. Decision making. Okay. Emotion detection. Okay. States of consciousness. Okay. Dreamers versus non-dreamers. Okay. And the olfactory episodic memory. Okay. Too bad. I was excited about the last one, but that's okay. Okay, so I think the decision making, definitely, and emotion detection and states of consciousness, these 3 seem to attract most. So I'll see how much I can go through these. Just bear with me. Decision making. Okay. Okay. So the decision-making example that I'm going to show you, this is work led by Thomas, Ph.D. in my lab, co-supervised with Pierre Ranville. And Thomas has worked on this intracranial EEG dataset. So these are, what you see here are the location of the electrodes across the 6 participants. These are intracranial recordings. So the participants had electrodes within the brain. This was obviously not volunteering participants. These participants had epilepsy, drug-resistant epilepsy, for which surgery has been recommended. And so they come into the hospital, they have surgery, they have the electrodes within the brain, and they spend about a week or 2 at the hospital. The idea is for them to have as many epileptic seizures as possible during that time, because they are recorded. So then we can look at the traces and identify where did the seizure start, and that's going to help the clinical staff decide on what has to happen on the surgery when they're going to remove a little part of the brain, and hopefully they get the seizure onset zone. And then that's going to reduce or actually stop the seizures occurring. But during this week or two, in many cases we have access to these populations if they agree, and we can run some experiments with them, and then we can collect data and analyze it. So what you see over here is just a plot in terms of number of electrodes to show you the sampling we got of the brain across the 6 participants. So we don't have all brain areas covered, of course, because this depends on the application. I'm going to go back first. I'm going to explain quickly the task. We have 3 conditions. The reason we call this a decision task, so basically the participants are going to be moving their eyes. Okay, so it's a saccade task or saccade task. After the first fixation, they are presented either with a diamond or with an arrow or with a square. Okay, these are the 3 conditions. If it's a diamond, it tells them, okay, it's going to be free. You can choose, you can decide on the direction of the eye movement, but still wait until the go signal, and at the go signal, they will actually perform the eye movement. Okay. In the case of the instructed saccade, which is this one here, they are given, the arrow tells them, okay, when the go signal comes after the delay period, then you have to shift your gaze, so you move your eyes either left or right, okay, depending on the direction of the arrow. And in the last condition, the control condition, a square comes up, and so this tells them, tough luck, we will not tell you anything, you'll have to wait until the end of the delay period, and then you'll discover right away where you need to do the saccade, and you do it. Okay, so it's a standard, typical delayed motor task. In this case, it's oculomotor task. What else do you need to know? I think that's all you need to. Oh yeah, the delay period is between 3.5 seconds to 10 seconds. So the delay period is quite long. Okay. And so the question that we were asking here is, if we look at the intracranial recordings, if we look at the signals we have across here, how do they differ across these 3 conditions? Okay, we're not looking. We did this in another study. No, we're not doing this here. We're not looking at can we decode the actual direction of the eye movement in the decision. We're just trying to see what separates these 2. What is different in my mind when I'm making a decision of where I'm going to move my eyes, compared to when I'm just told I will have to move my eyes right or left after at the go cue. Okay. So what we get, something like this. Again, these are illustrations to show you how we're using the machine learning in this type of context. The numbers here are decoding accuracies of the classifier. I think in this case here it was an SVM classifier. And so this tells you over time, going up to 2 seconds, the areas that light up in red are areas where the decoder gave us significantly, statistically significant decoding accuracies, up to, like you can see here, up to like 90% or 80%, when comparing the free to the baseline, just before the beginning of the task. So when do brain activations differ from pre-stimulus activity? Okay, and where do they happen to be? The second one here is instructed versus baseline. This is where they did not make the decision condition. It's actually they were just given the instruction. And the third one was the control condition, where there was no information provided. And so, to cut a long story short, the interesting part here that I can draw your attention to is the fact that if you compare these 2, the first and the second line, there is more activity going on later in time than here. There is more, and again, I did not mention this is high gamma power. So we're looking at the activity in the high gamma frequency, and we see that it lasts longer for the decision making compared to the instructed, where actually the information is self-sufficient. You see the arrow, you know what you're going to do. The activity of gamma dies out quite quickly. But when you're making that decision, you have sustained gamma activity in prefrontal areas, including DLPFC. You can track the time. Here, what you're plotting on the brain is the time of maximum decoding accuracy of your classifier. So you can plot across the brain what was the time from onset where you had the best decoding accuracy. And this again shows you here, when you see the yellow up here, that in these prefrontal areas, the best difference between baseline activity and the decision-related power in the gamma range was around 1.5 or 1.2 seconds. So this activity happens later. If I show you this very briefly, we can do this across all frequency bands. I'm not going to go into details. What gamma activity is doing most of the time, it goes up and down in the case of instructed, and in free it goes less up, but then it continues to be sustained, whereas it dies out very quickly in the case of an instructed saccade. In other words, meaning that probably this gamma activity, this sustained gamma activity, is important for the deliberation and for the visual-motor transformation related to taking your own decisions. Okay? I'm going to wrap this one up very quickly. You can do a classical conjunction analysis, but now it's again based on decoding accuracy. So this is decoding accuracy for free larger than control. This is free larger than instructed. And you can do conjunction analysis, and this tells you what are the areas that are really specific to decision making in this task. This is intracranial EEG. So we have very high signal to noise ratio, high temporal resolution, but also spatial. And so you can look at single trial activations. These look very similar to raster plots for those of you who are familiar with electrophysiological recordings, for example, in primates. So across here, you see these are trials. So each single trial, you can look at the gamma power. And what you see here is the onset of the saccade. And so you see, just to give you an idea, is that for the control condition where there was no information about the direction before, there is gamma activity that comes just before the onset, follows the onset of the saccade, of the eye movements at the go signal. Whereas for instructed and free, we don't see this. In other words, there is some processing that had to occur only once. It was done already in the delay period. So for these at execution, you did not need this gamma activity. So this pinpoints to this gamma activity in the medial frontal gyrus that is specific to oculomotor activity. I can go into more details, but again, I'm not going to do this because the aim was to just give you some examples of how this works. You can look across time. What you see in all these figures just below here, this is decoding accuracy of your classifier. This tells you your two conditions, free and control. Over time, all of this is 2 seconds, and you can pinpoint in terms of milliseconds where does the decoder classify these two tasks, and you can do that also in single trials. Okay. I'm going to show you briefly the decoding the emotion with intracranial EEG again. These are across 11 participants. These are recording sites. This is data that was collected in Lyon, in France. And what the participants saw was a set of faces. And what they had to detect was, I think, the fearful face. That was the target. And this is just a very brief example. If you take the activity in one of the recording sites and you look for joy, neutral, anger, scramble, we just scramble the faces, and fear, you can do a time frequency map because this is, again, electrophysiology, and you can see over time and frequencies what's different across these different conditions. And you see here that for fear, this elicits a strong gamma activity. And obviously you can use this as features for your decoder. So if you train your decoder, on this, and the reason I'm showing you this is not to give you all the details of the experiment, but just to show you how you could potentially use decoding algorithm for your data. In this case, what you see here, again, this is a time frequency map, like the ones you see down here. So in time, what is the frequency content of your EEG signal? But here, what you can do is you can actually represent decoding accuracies across the time frequency space. And so you can identify that in the gamma range, there is high decoding between the two conditions. Do I have time for the unconsciousness? No, maybe not. Huh? Okay, well, you can talk to me about any of the other studies that you were interested in, I think, right? Because that way we can en termine à l'heure, sinon.
Because of the lunch.
Yeah. Okay. So for those of you who are interested in any of the other studies, I'll be around a little bit more, but you can also get in touch with me, I think later. Maybe I wanted to finish off with just this. Where is it? Okay. Okay. So applying machine learning to electrophysiological brain signals such as EEG or MEG is a promising data mining tool for systems cognitive clinical neuroscience. An important point that I'd like to make is that if you have a priori knowledge, you should probably use it. Because there's a lot of buzz around AI in general, a lot of buzz around deep learning also in general. It is useful for many things, but I think we just always need to remind ourselves, I'm going to do this task. Before I jump straight to the deep learning, do I really need deep learning for this type of task? In many cases, yes. In many other cases, it's not at all the case. So I think this needs to be seen more critically in terms of, do I really need a big hammer for this problem, or am I completely going to be on the wrong path. This is the final announcement. We will be running again, Main this year, 2019. The dates, here we said November 2019. It's probably going to be 14th to 17th, but don't tell anybody I told you this, because we still need confirmation from CRM and so on, but probably somewhere between 14th and 17th, it might change a bit. This is why, for now, just keep in mind, it will be November, so between SFN and NeurIPS. And yeah, I think c'est tout ce que je voulais vous dire. If you have questions, you can also ask me questions, French and English, comme vous voulez. Merci.
Thanks so much for the lovely introduction to machine learning. I hope it was useful for many of you. We have a few minutes for quick questions before we run for lunch.
I was wondering if you'd be willing to share your slides, because there's so many studies you didn't have time to cover, and they all sound super interesting.
Yeah, so absolutely. I mean, some of the results are not published, so that's the only. So for those that are not published, I can't share them yet. But for a lot of the introduction to machine learning, that's easy, of course. I can share those, and some of the studies are published, so I can also share those. But I'll be happy also to walk you through any of the results that you were interested in.
We can post it on the Slack. Maybe remove the slides.
Yeah, absolutely. I guess it's time for lunch.
Yeah, I guess we're running late for that. Thanks
Okay.
again, Jack.
Merci.